This is Chapter 3, Module 3, Cellular Transport, Part 2. In Chapter 3, Module 3, Cellular Transport, Part 1, we describe the different modes of transport, including simple diffusion and osmosis. In Part 2, we will describe carrier-mediated transport and vesicular transport. Some ions and organic molecules are able to pass across the plasma membrane through transport proteins. When this happens, it is called carrier-mediated transport because a protein helped or carried the molecule across. Carrier-mediated transport has very specific characteristics. First, the transport proteins are very specific. Each transport protein accepts one type of molecule to be carried across. Second, the rate of transport can only be as fast as the transport protein can carry them. This is called a saturation limit of the transport protein. And third, cofactors are often needed to trigger the transport protein to carry the molecules. These cofactors are such things as hormones. Most of the time, one substance will move in one direction using transport proteins, but transport proteins can also carry two substances across the membrane at the same time. If two substances move in the same direction at the same time, it is called cotransport. If one substance moves in while the other substance moves out, this is called countertransport. There are two types of carrier-mediated transport, facilitated diffusion or active transport. Facilitated diffusion is a passive process where carrier proteins transport molecules that are too large to fit through the protein channels. Glucose and amino acids are examples of molecules that are too large to fit through these channel proteins, so they rely on transport molecules to get them across the plasma membrane. Glucose or amino acid molecules bind to receptor cells on the carrier protein that is embedded in the plasma membrane. This connection between the molecules and the receptor sites cause the transport protein to change shape, and the molecules pass through to enter the cell. The receptor site is specific to certain molecules and will only change shape when the specific molecule binds to its complementary protein. This is a passive process because it doesn't require ATP or energy. However, it is facilitated by the protein changing shape, so this carrier-mediated transport called facilitated diffusion is a passive process where molecules will diffuse across the plasma membrane from high to low concentration with the help of a transport protein. Active transport is a carrier-mediated transport process that requires ATP. In this type of transport, molecules are moved against their concentration gradient. That is, they need to be pumped from an area of low concentration to an area of high concentration. It is the opposite movement of what the molecules would do in diffusion. Ion pumps will pump ions such as hydrogen, sodium, potassium, calcium, and magnesium against their concentration gradient. An example of an active transport is the sodium-potassium exchange pump. During this active transport, sodium ions are pumped out of the cell against their concentration gradient, and potassium ions are pumped into the cell against their concentration gradients. This process takes energy in the form of ATP. Every 1 ATP will move 3 sodium out of the cell and 2 potassium into the cell. The sodium-potassium exchange pump is very useful in maintaining the concentration gradients of these two ions. Another type of active transport is vesicular transport. In this type of transport, materials move in or out of the cell in vesicles. Vesicular transport uses ATP. When vesicles carry materials into the cell, it's called endocytosis. There are 3 types of endocytosis: receptor-mediated, pinocytosis, and phagocytosis. Receptor-mediated endocytosis is when receptors on the cell membrane bind to target molecules in the extracellular fluid. These extracellular molecules are called ligands. When ligands bind to the receptors on the cell membrane, the membrane begins to form a dimple, which gets bigger and finally pinches off inside the cell, carrying the ligands within it. This newly formed vesicle is called an endosome. Another form of endocytosis is called pinocytosis. Pinocytosis is when the endosome that is formed carries extracellular fluid instead of the ligands. This endosome brings in extracellular fluid into the cell, and one could say that the pinocytosis is how the cell drinks, by bringing in fluid. Another type of endocytosis is called phagocytosis, which is cell eating, where the cell engulfs large objects and brings them into the cell, forming phagosomes. These large objects, such as bacteria, will be digested in the phagosome and then released. The cell membrane will change shape in order to engulf large objects from the extracellular fluid. The cell membrane elongates, forming pseudopodia that reach around the object, such as a bacteria, and brings it into the cell in the phagosome to be digested. Watch as this white blood cell chases a bacteria and forms pseudopodia, which will engulf the bacteria and bring it inside the cell.
In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Exocytosis is the process of using vesicles to release material outside of the cell. In exocytosis, the vesicle fuses with the cell membrane and opens up on the extracellular side of the cell. The cell will release secretions that were produced in the cell, such as hormones or neurotransmitters. The cell will also release waste products, such as the waste product created from the digestion of bacteria within the phagosome.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
This ends Module 3 of Chapter 3, Cellular Transport.